Hello friends, welcome to SR Academy.
Welcome to SR Academy. Empowering minds, igniting curiosity. Your gateway to knowledge unleashed. Let's get started.
Hello everyone, welcome to my YouTube channel. So today we are going to discuss about the current scope of development of tissue engineering. So previously we discussed about the introduction, then we discussed about traits, tissue triad, and then the next topic we are having is current scope of development. Current scope of development is in the sense where exactly tissue engineering stands as of now, whether it has been used in the hospitals or to treat the patients, or it is still under preclinical trials or clinical trials. Now, since this subject is there for biotechnology students, I don't have to explain what is preclinical, clinical, all those things, right? I hope all of you understood. Preclinical trials is nothing but whatever you are going to develop the new technology, new drugs, whatever it is, we have to test it on animals first. For that is called as preclinical trials. And if it is approved by these particular regulatory bodies like FDA in US, then or different approval by regulatory bodies from Europe and Japan and all, then it will be, the license will be given to test it on the humans. That is called as clinical trials. So testing on animals is pre-clinical trials, testing on humans is clinical trials. So most of the researchers research and development with respect to the tissue engineering, it's still in preclinical and as well as clinical trials, okay, which is not completely widely used in patient treatment, okay, and but still there are a lot of things, or in few applications at smaller scale we will be using the tissue engineering. So in the particular treatment applications, because in order to get the particular tissue engineering to be used in patient's treatment, that requires a lot of effort. I mean, what do you say, a lot of research and development should occur, optimization should be there, and there are plenty of parameters will come when you are implanting a particular thing inside the body. So there are a lot of parameters come that need to be studied, and that need to be optimized, that needs to be studied on a group of people, then a different set of groups of people with different ages, different all these things. So that is a very tedious job and it requires time. So at present, let's see what is the current scope of development of tissue engineering as of now. So as of now, at present, the tissue engineering plays a relatively small role in patient treatment. That means it is not playing a major role in the patient treatment, but still it is playing a small role, where we cannot say that there is no application, there is no application as such currently from the tissue engineering in real time with the patients and all. There is a small role which from the tissue engineering that is used in patient's treatment. Okay, so for example, we are having the artificial skin, valves, cartilage. These are some of the examples which are engineered by the tissue engineering, or engineered tissues, what you can call, that have been already approved by the FDA. So artificial skin for the skin grafts or the skin burns, then valves like for blood capillaries and for heart valves, and then cartilage. So cartilage is also which is where the joints and all it will be used. So these are the three things which has already been approved by this particular FDA, okay, regulatory body, okay. So however, currently they have limited use in the humans, okay, very less or limited. So supplemental bladders, for example, then small arteries, okay, small arteries in these things, particular human heart, then skin grafts, okay, skin grafts what I just told you, then cartilage I already mentioned, and even full trachea that have been implanted in the patients, okay, the full trachea, okay, the trachea has been implanted in the patients, but the procedures are still experimental and very costly. That's why we cannot afford, one cannot afford all these things, because it's very costly. Why costly? Because it is not widely in use as of now. So all these things have been done. See, the supplemental bladder, replacing the small arteries, skin grafts, cartilage, these all are not replacing the whole trachea. These are not the small things, okay. All these things plays a very important role, but it has got limitations, like for example they're under still experimental, that is clinical study, and very costly also. So while more complex organs like tissues, like for example heart, lung and all, these all tissues, small cells had been is still under procedure, that this everything will be there, and in future we can expect these things to become faster. But when you are talking about the complex organs like heart, lung, liver, these tissues has been successfully recreated in the lab. That means in vitro, by using tissue engineering, the heart has been developed, lung has been developed, liver has been developed. But they are so long, or you can say they are a long way from being fully reproducible. That means, and ready to implant into the patient. That means it takes it still requires a lot of effort, research and development to understand more deeper aspects of these particular organs, so that it can be used completely, it can be used for organ replacement or direct implant into the patients. So this requires a lot of studies, again parameters, because these are highly complex organs, in the sense there are a lot of lot of things need to be understood, because nervous system, how exactly it is working with the heart cells and heartbeat, then lung also, okay, all the dendrites are there, and in between whatever the oxygen and this exchange, gas exchange, all those things need to be studied, and liver also. These are all complex organs, but they have been synthesized in the lab. See, the beauty of the tissue engineering is that now in vitro we are having a ready heart, lung and liver, but only we need to implant into the patients or the human beings. So it takes a lot of time, but as of now we have heart, lung and liver which is being cloned, or is being generated or grown in the laboratory by using tissue engineering. What we can do, how it will be help us. So if imagine you have got a particular heart disease, imagine you have to develop a new drug for it. Okay, so for that what you have to do, you have to test it on a particular heart. Suddenly you cannot test it on the animals. We will imagine that testing on animals, obviously we will end up killing animals. Definitely if the drug reaction and that, this and all, there is a maximum probability that animal might get heart related diseases also if it is not dying. So in order to avoid these particular things, if any new drug has been developed for a heart or a lung or a tissue or a particular liver, then I can have the clone, I can test it on the heart which is being generated by the tissue engineering in the laboratory. Understanding? Lung also you can test, liver also you can test. Understanding? So for this particular application it will be helpful. Tissue engineering will be helpful. So these tissues, however, can be quite useful in research, and especially in drug development and all, wherever you need to use the new drug to develop the particular a particular drug, and how it is going to be useful as a to treat a particular disease which is related to heart, liver and lung. That can be understood, that can be tried on this particular at a laboratory scale only. So using the functioning human tissue to help screen medications, my medication candidate could speed up the development and as well as provide a key tools for facilitating personalized medicines while saving the money and reducing the number of animals used for the research. So this is what we can do with the available the tissue engineering products. For example, using these all which is being done in the laboratory, so we can have the functioning human tissue, okay, to help screening. So we can screen the medication candidates, that means the drugs, whatever the new drugs has been developed, that we can screen. That means whether they are safe or not. Okay, and also it will speed up the development of this particular drug. So for example, if you are trying, imagine I have developed a new drug and I am trying it on a human being. So to get to know how exactly it is functioning on the heart, I have to take him to the every checkup, okay, after frequently, like after month or a weeks and all. So here if you are having the heart separately outside of the body, so you can check faster, you can do as many as n number of trials, okay, at a same time you can have around 10 hearts. So with 10 different concentration of the drug, you try it on, try on these particular hearts, see which is giving better results, which is curing the disease faster. So that can speed up the development, and also it will provide the key tools for facilitating the personalized medicines also, so that it can facilitate the personalized medicine. You don't have to require for this particular, every time go to the doctor and all, while saving money, and as well as reducing the number of the animals which is used for the researches. So these are some of the applications, or even though it is not being widely used in the particular patient treatment and all, but whatever has been developed in the laboratory, that can be useful to check the new drugs, or to check any new kind of research and development which need to be tested on human beings. You can directly test it on those particular organs and check whether how they are responding, so that we can avoid killing animals, and you can avoid the time which is being consumed, and also the adverse reactions on human beings. All those things can be avoided, so that it will be more safer. Okay, so more safer preclinical or more clinical studies can be done with the help of tissue engineering. The next topic we are having the use in therapeutics, cells as therapeutic agents, that we will discuss in the next class.